Hello and welcome to this session, Learning Outcome LO2. Describe the main features of eukaryotic membrane structure. Today, we will learn how eukaryotic cells manage to control their internal environment so effectively. The key lies in the intricate design of their membranes. In this session, we will explore the main features of eukaryotic membrane structure and understand their essential roles in maintaining cellular function and homeostasis. The foundation of eukaryotic membranes is the phospholipid bilayer. Each phospholipid has a hydrophilic head and hydrophobic tails. They arrange into two layers, creating a stable barrier. This barrier is semi-permeable, allowing small nonpolar molecules to pass. It restricts ions and larger molecules, maintaining cell integrity. Proteins embedded in the bilayer give membranes their functionality. Intrinsic proteins span the membrane, aiding in transport. Extrinsic proteins sit on the surface, often involved in recognition. These proteins support communication and structural stability. They are crucial for chemical reactions and transport processes. Cholesterol molecules are scattered throughout animal cell membranes. They fit between phospholipid tails, regulating fluidity. Cholesterol prevents membranes from becoming too rigid or too fluid. It adds stability and decreases permeability to small molecules. Plant cells use other sterols for similar roles. Glycoproteins and glycolipids are found on the outer membrane surface. They have carbohydrate chains attached. These structures aid in cell recognition and signaling. They also play roles in adhesion and forming tissues. The glycocalyx serves as a protective layer around the cell. The fluid mosaic model describes membrane behavior. Fluid refers to phospholipid and protein movement. Mosaic reflects the protein patchwork in the bilayer. This model shows membranes as dynamic and adaptable. It highlights the constant adjustment to cellular needs. Eukaryotic membranes allow lipid-soluble molecules to diffuse freely. Protein channels help move ions and glucose. Active transport and diffusion facilitate complex transport. Exocytosis and endocytosis manage large molecules. This selective permeability is crucial for cellular function. The phospholipid bilayer forms the membrane's basic structure. It creates a semi-permeable barrier, controlling the cell's environment. Its fluid nature allows flexibility and shape change. Lateral movement of molecules maintains even distribution. This bilayer is essential for processes like endocytosis. Intrinsic proteins span across the bilayer. They facilitate transport of ions and glucose. Active transport pumps use energy to move substances. Some intrinsic proteins act as receptors. They are vital for cellular communication and signal reception. Extrinsic proteins sit on the membrane surface. They aid in cell signaling and communication. These proteins provide structural support. They anchor the membrane to the cytoskeleton. Some extrinsic proteins catalyze reactions on the membrane. Cholesterol fits between phospholipids and membranes. It regulates fluidity in varying temperatures. Cholesterol adds stability by reducing phospholipid movement. It decreases permeability to ions. This helps prevent ion leakage, maintaining cellular function. Glycoproteins and glycolipids have carbohydrate chains. They assist in cell recognition by the immune system. These structures are receptors for hormones. They enable cell adhesion, forming tissues. The glycocalyx layer protects the cell from damage. The fluid aspect is vital for membrane repair. It allows proteins to move to needed areas. Vesicle formation occurs during endocytosis. Efficient diffusion within the membrane is essential. This fluidity supports dynamic cellular processes. Membrane fluidity allows for quick repair. Damage can be efficiently managed. Proteins and lipids move to cover breaches. This ensures continued cellular protection. Fast repair is crucial for cell survival. Proteins move within the membrane to where they are needed. This movement is facilitated by the fluid nature. It ensures functional distribution. Proteins can relocate for optimal cellular response. This adaptability is key for cell operations. Vesicles form during endocytosis and exocytosis. This process is supported by membrane fluidity. Vesicles transport materials in and out of the cell. They play a role in nutrient uptake and waste removal. Fluidity ensures efficient vesicle formation. Fluid membranes enable efficient diffusion. Substances move swiftly across the membrane. This is crucial for nutrient and waste exchange. Fluidity supports even distribution of molecules. Efficient diffusion is vital for cellular health. Membranes are not rigid structures. They constantly adapt to cellular needs. The fluid mosaic model explains this dynamic nature. Proteins and lipids adjust for optimal function. This adaptability is essential for cellular processes. Membranes adjust to environmental changes. This adaptability ensures cell survival. Proteins and lipids shift for optimal performance. Membrane dynamics support various cellular functions. Adaptability is a key feature of eukaryotic membranes. Eukaryotic membrane structure is crucial for cell function. It controls the internal environment. Membranes support communication and transport. Their dynamic nature is key for adaptability. Understanding this structure reveals the complexity of cellular operations. In conclusion, eukaryotic membranes are highly complex and dynamic structures, intricately designed to support vital cellular functions. Gaining insight into their organization and behavior deepens our appreciation of the sophistication and intricacy of cellular life. If you need any help, please reach out via WhatsApp at 07484361688. Our team is ready to assist you. Thank you, and see you next time.